If you remember from our last topic, we have two broad groups of true animals. The true animals are known as the eumetazoans, and they're divided into the deuterostomes. That would include us, and that's about 10% of true animals. And then we have the non-deuterostomes, about 90% of the true animals. Hey, the math works here. In addition to the true animals, the eumetazoans, we have what are called the parazoans. Parazoans are almost animals. They're on the cusp. They're remnants of the very early ancestors of animals. Today we're going to talk about the parazoa and the eumetazoa. We're going to focus on the non-deuterostomes. The non-deuterostomes are all invertebrates. Invertebrate is an informal name for an animal that lacks a vertebral column. It lacks a backbone or spinal column. There are a few deuterostomes that are also invertebrates, and of course, depending on who you talk to, the sponges may or may not be considered animals. We're going to consider them invertebrates as well. The Animalia is a fascinating kingdom, but we're going to focus on what I've drawn in green boxes here. Those are the groups that you should know for your final exam. Now, we've seen this figure before, but let's go over it again quickly. Remember that we start out at the root of this phylogenetic tree with our ancestral colonial choanoflagellate. A choanoflagellate is a single-celled organism that has a flagellum that creates a current of water, and at the base of the flagellum there is a sticky collar that traps food. These cells can exist as a colony. So a bunch of cells that are working together, but the cells are not specialized in any way. This ancestral choanoflagellate gave rise to the parazoa. Again, para means next to, zoa means animal, so they're next to the animals. They're almost animals, but maybe not quite. These are the porifera. Porifera literally means full of holes. And that's a good way to define a sponge. It's a thing with a lot of holes in it. Then we have the eumetazoa, the true metazoans, the true animals. They diverged into the radiata, which includes the cnidaria, another group called the ctenophora, which we're not going to focus on, although they are quite fascinating. And we also have the bilateria. The bilateria are bilaterally symmetrical. And the bilateria diverged into the protostomes and the deuterostomes. Let's start with a quick look at the parazoa, the phylum Porifera, the sponges. Now, this is not a real sponge. This is a synthetic sponge, and these are the sponges that you see most often today, which is a good thing because we're not harvesting these living organisms from the ocean. But this is what a real sponge looks like. And if you go back many decades, this is what most people were using as a bath sponge. But this is produced by living organisms. This is made mostly of collagen, which is a very flexible, elastic protein, and it's produced by a colony of little tiny organisms that look a whole lot like choanoflagellates. Again, the term porifera means pore bearer or full of holes, and that's what sponges are. They're full of holes. They are very closely related to the choanoflagellates, which are considered a protistan group. So the cells that make up the majority of a sponge look identical to those choanoflagellates. They have cells that are arranged in very loose aggregations. In fact, what you can do is you can take a sponge and you can put it in a blender, blend it up, pour it into an aquarium, and those cells will come back together and form new sponges. They have very limited cell specialization and no true tissues, and this is the reason that they're considered parazoans and not eumetazoans. They come in a lot of different forms. They're sessile, which is a fancy way of saying they don't move around, they stay stuck to something. And we have a lot of varieties. We have at least 100 freshwater varieties, but the vast majority are marine. They live in the ocean. They are filter feeders. Now notice that these examples here have a hole at the top. They're shaped like a tube. What they do is they pull in water and then they filter bacteria and other small life forms out of the water. That's what they feed on. So they create a current of water using the flagella on each of those choanoflagellate-like cells. They do not have any nervous tissue, certainly nothing like a brain, but not even simple nerve cells, and they do not have any muscle cells. They have a very simple body plan, which is basically a sac through which the water is drawn in and then pumped out, again using these flagella. Here is a typical sponge. Now some sponges are hard and scratchy, and some are quite soft. This is one of the hard ones. If you stepped on this with bare feet, it would hurt. So here we have a sponge. It's got two main layers to it. So it has an epidermal layer on the outside. It's a hollow cylinder, and on the inside it has a layer of choanocytes, which are very similar, pretty much identical to choanoflagellates. The choanocytes have a flagellum, and that creates a current of water. And they also have this sticky collar that bacteria and algae and so on in the water can stick to. Now we do have two more cell types. We have what are called porocytes that create pores, little holes in the wall of the sponge. And then we have cells called amoebocytes, which can move around freely. So what happens is these flagella are beating like crazy on the inside of the sponge, and they're creating a flow of water out the top of the sponge. There's an opening at the top. Water gets drawn in through these pores, and food is trapped by the choanocytes. The amoebocytes wander around, and they take a little bit of food from the choanocytes to feed themselves. And if some of the choanocytes aren't getting enough food, the amoebocytes will transfer food to them. The amoebocytes are wandering through this layer of goo. So between the epidermis and the choanocytes, there's something called mesohyl, which is just a layer of goo. It's just a layer of gooey proteins. Embedded in that layer of goo are structures known as spicules. Now, these can be made of calcium carbonate and other minerals that are quite tough, but if we're dealing with a soft sponge, like the kind you might use in a bathtub, then we don't have these hard spicules. Instead, we have a more kind of ambiguous, vague sort of structure where instead of these hard, scratchy spicules, we have collagen. So in the soft sponges, we have lots of collagen just arranged in a very open network. Think of SpongeBob, perhaps, where water is just sucked in and then distributed through the holes and the pores of that sponge. Another view of a typical sponge here, again, water being sucked in through pores and then expelled out the top. Those amoebozoa that wander around in the goo between the two layers will occasionally undergo meiosis. They're the only cells that do, and they produce sperm and egg, and they just dump them into this cavity and they get expelled out into the water. Sponges are hermaphrodites. So those amoebozoa will produce both male gametes and female gametes. And sponges do come in lots of forms. Again, some of them are rubbery, some of them are very soft, some of them are very hard and scratchy. And it just depends what those spicules are made of. Again, the spicules are the little bits that are embedded within the goo. Are they made of collagen? If so, you'll get a soft, spongy sponge. If not, then you'll get a hard, scratchy sponge. And finally, a couple more examples here. Again, some soft sponges like the ones you see on the left, and then the ones on the right are quite hard. You would not want to take them into a bathtub with you. The spicules there are made up of calcium carbonate. The next group we're going to talk about are the cnidarians. Notice that the C is silent. These are eumetazoans. Remember that eumetazoans have true tissues. This is what distinguishes them from the parazoans, the peripera. So everything but the sponges are considered eumetazoans. The cnidarians come in two body shapes. We have what's referred to as a medusa, that's the jellyfish form, and then we have what's referred to as a polyp, that's the sea anemone form. But this group includes not just jellyfish and anemones, it includes coral. So if you have a close look at a piece of coral, it's actually made up of hundreds or thousands of little tiny polyps, little tiny animals. And we have things like hydras. This is a group that includes some freshwater members that take the polyp form. So some cnidarians will alternate between a medusa and a polyp. Some are purely medusa, some are purely polyps. There are at least 10,000 species. Most of them live in the ocean, but there are some freshwater forms, and you can find some of those in lakes and ponds where we are. So here's a typical medusa form, a jellyfish, and here's a typical anemone form, a polyp. The cnidaria are very simple. They are true animals, but they're the simplest of the true animals. They have two tissue layers, and those tissue layers are derived from the two germ layers that they began with. So remember, we start out as a ball of cells. One side gets pushed in, that gives rise to a gastrula. In us, there's a third layer that develops in between those two layers. That's the mesoderm, and we become rather complex organisms. In the case of the cnidaria, they have these two germ layers, and they don't really develop beyond that. So they have a simple body plan. They have a gastrovascular cavity that serves as the stomach, but they do not have a separate anus. So the mouth also serves as the anus. The gastrovascular tissue is going to secrete enzymes that break stuff down in that cavity, but then the wastes have to be released through the same opening they came in through. Because there's two cell layers, we call them diploblastic, so di meaning two. They have the epidermis that develops from the ectoderm. They have the gastrodermis that develops from the endoderm. There is no mesoderm or anything derived from mesoderm. Here we have a rather classic cnidarian that alternates between a medusa form and a polyp form. So again, the medusa is shaped like a jellyfish, and the polyp is shaped like a sea anemone. This is a colonial animal, which means we have lots of little tiny animals that are all stuck together. You can see that on the left. Now notice that their stomachs, their gastrovascular cavities, are stuck together. They don't have a circulatory system. Instead, they share food within this gastrovascular cavity. So if one of these little sea anemone like polyps catches food, but the one next to it doesn't, they share food. So we have what are called gastrozooids. Don't worry too much about that name, but they catch food. That is their function. And then we have these gonozooids. Again, don't worry about the name there, but what they do is they produce medusa. They release these little jellyfish. The jellyfish go off, and the jellyfish produce sperm and egg. So it's the medusa that are having sex. The sperm and the egg come together. That forms a zygote that develops into a blastula, and that develops into a larva that can swim around. The larva will find a place to settle down and give rise to a new colony. Now, this alternation between the polyp and the medusa, don't get that mixed up with the alternation of generations in plants. Remember, in plants, we alternate between a diploid sporophyte and a haploid gametophyte. That's not happening here. Instead, we just have these two diploid stages. The polyp is focused on eating. The medusa is focused on sex. Now, what we'll see in the other cnidarians is that some of them have specialized. Some of them have given up the medusa stage. They're polyps all the time. That would be the sea anemones. And some have given up the polyp stage, and they're medusa all the time. That would be the jellyfishes. But some of them still do this whole cycle. This group is mainly marine, but there are a few freshwater species. There's 3 classes. So we have the hydrozoa, and they alternate between a medusa and polyp form, as we've talked about already. We have the scyphozoa, and they strongly favor the medusa, the jellyfish form. And then we also have the anthozoa, which strongly favor the polyp form. And this would include things like corals and sea anemones. Some cnidarians live in fresh water, and this is one known as hydra that you can find locally. If you go to a local pond or lake and maybe look on the underside of a pond lily, so those are the leaves that float on the surface of the water, you might see one of these guys. They have tentacles that they use to catch food, and they have an interesting way of reproducing. They can reproduce asexually, and you're seeing that on the left here. You're seeing a new individual that is just budding off of the parent. Imagine if we could reproduce this way. Imagine if you could just kind of bud off a new human from your hip. That's what's happening, and that's what they do through most of the year when conditions are good. In the fall, when the lake is getting cold and it's about to freeze over, they produce sperm and egg, and they produce these zygotes that will then survive through the winter and hatch out as adults later, and that's what you're seeing on the right. Here's some more examples of polyps, some very common sea anemones. Again, what happens in their development is that we have a blastula. It gets pushed in on one side, and that forms a blastopore. The blastopore becomes the mouth, but it also serves as the anus. They don't develop a second hole. So imagine if we lived that way. We would take in food through this one opening, we would digest it, and we would spread that food to all the cells, and then we would take the waste and we would expel it through the same opening. Kind of gross. Here's another form of marine cnidarian. This is something known as a sea pen, and you can see that we have a bunch of individual animals. So this is colonial again. Different from a sponge. A sponge is a colony of single cells. This is a colony of separate animals. And finally, we have coral. If you take a very close look at coral, like you're seeing in the bottom left, you'll notice in living coral that there's lots of tiny little polyps. So there's hundreds or thousands of individual little animals, and they secrete calcium carbonate, which makes the hard part of the coral. There's a lot of other cnidarians that work this way. Portuguese man-of-war, which is a big jellyfish, is actually not one animal. It's a floating colony of animals. So we've looked at diploblastic animals, and the next step would be to look at triploblastic animals. And all of the animals we'll look at from here on out are triploblastic. Tri meaning three, so they have three tissue layers in the early embryo. We have a mesoderm now. Mesoderm gives rise to muscle. It gives rise to connective tissue, and in us that would include the skeleton. Connective tissue is the tissue that binds all the other tissues together. And first we'll talk about triploblastic animals that lack a coelom. They lack a body cavity, so they're solid basically. So we have the platyhelminthes. Helminthus means worm. Platy means flat. Think of platypus. It's got a flat puss. It's got a flat bill. So flatworms are the platyhelminthes. We have several different groups included in here, and I'm not going to talk about the Lophotrochozoa, so we can ignore that. But we're going to talk about the platyhelminthes, and these are the flatworms exactly like what they sound. They are worms that are flattened. We have flukes and we have tapeworms. So the flatworms are free-living, many of them. Flukes and tapeworms are parasitic. Here's an example of a flatworm, a free-living marine flatworm. Some of them can get quite large and some of them are quite colorful. There's at least 20,000 different species. They're found in marine environments and in aquatic environments. You can find them in ponds and rivers around here. You can find them in soil as well. As mentioned, many of them are free living, but there are some parasitic forms. They're all bilaterally symmetrical. So this is a change from the cnidaria, which were radially symmetrical. They're triploblastic, so they have three germ layers. They have the ectoderm, the mesoderm, and the endoderm. And they have true tissues. Because they have the mesoderm, they develop connective tissue and they develop true muscles. So let's take a look at the different groups here. We have the Turbellaria, and I don't want you to know this much detail, but this is a free living group, marine and aquatic. They're predatory. So we do have some in ponds and rivers. They're usually a centimeter or two in size, and they'll hunt little tiny crustaceans and other things in the water. They do not have a circulatory system. They do not have a respiratory system. So even though they are multicellular and they do have well-defined tissues, they lack a lot of the more complex organ systems that we quite often associate with animals. They rely on diffusion. They are quite flat, and the reason they're flat is that increases the rate at which they can take up materials from their surrounding, including oxygen. They have cilia on the underside, the ventral surface, and they secrete mucus. And basically they secrete kind of a little slime layer, and then the cilia are used to move along that. They have cephalization. Now that's a term I haven't used yet. Cephalo means head. Cephalization is the development of a head. And with animals, if something has a head, and the head just has to be a place where there's a couple of sensory organs stuck, that makes the animal bilaterally symmetrical. So the act of having a head makes you bilaterally symmetrical. They can reproduce asexually through regeneration. So some of them can just split. Some of them you can take them and actually dice them up into little bits and they will regenerate. But they do undergo sexual reproduction as well, and most of these guys are hermaphrodites. Then we have the monogenea, which I'm not going to talk about, so I haven't highlighted them here, but these are parasites. They're all parasites of fish. An interesting group, but they're not something you're likely to run into. We have the trematodes, which are parasitic. They're known as flukes. They have suckers at the head end that they use to attach. There are some that will parasitize the liver, some the lungs, some the bloodstream. If you're taking microbiology next term, we'll talk about those in microbiology. But I'm not going to go over them in this course. They tend to have a really complex life cycle as well as most parasitic organisms do. The interesting thing about them, at least I find it interesting, is there's a large group, the schistosoma, that absolutely have to have a snail as an intermediate host. They tend to be rather picky about these kind of things. But again, the monogenea and the trematoda, we won't talk about them, but we'll move on to our last group here that we will talk about, the cestoidea. These are also parasitic. So we have the Turbularia, which are non-parasitic, free-living, and then we have the cestoidea, which are internal parasites. These are the tapeworms. So at one end, they have hooks and suckers that they use to attach to the inside of the intestine, and they feed on pre-digested food. We've talked about them before and the fact that they've become very, very simple during evolution. They have a thick epidermal covering, and the reason for that is to protect them against the acids and antibodies and the immune cells that are produced by your body. They do have sexual reproduction, but they're hermaphrodites, and in many cases they have sex with themselves. They have these segments called proglottids, and the proglottids only contain reproductive organs. There's nothing else going on in the proglottids other than reproduction. They have a very complex life cycle. Again, most parasites do, and they have an intermediate host as well as a definitive host, and we'll talk about what that means in just a second. The final host, the definitive host, is a vertebrate. So again, we have the phylum Platyhelminthes here, flatworms. The larger group, Lophotrochozoa, don't worry about that name, but it is a much larger group that includes a lot of interesting, almost entirely microscopic forms that, if we had more time, we could talk about. Anyway, we have the Turbularia, and if you just say free-living flatworms, that's fine. There's lots of species here. Some of them are really, really pretty. We have Dugezia. Again, don't worry about that name, but that's the one on the upper right. This is a very, very common freshwater flatworm. If you go and just take some sediment from the bottom of a pond, you're going to find some of these things. They're really neat. As I mentioned, you can actually chop them up in little pieces and they will regenerate the missing part. If you cut off the head, it will generate a tail. If you cut off the tail, it will generate a head. They're quite fascinating. The monogenea, again, we're not going to talk about, they're all parasitic and mostly parasitic of fish. The trematoda, flukes, we'll talk about those in microbiology, but I do want to talk again about the cestoidea, which are the tapeworms. They're quite fascinating. Looking at the free-living flatworms, this is the common body plan that they have. So this is Dugezia, a really, really common freshwater flatworm. We have a very, very simple nervous system. So we have ganglia at the front. Ganglia are just concentrations of nerve cells. So it's not really a brain, but it's kind of a brain. And then we have eye spots. And I mentioned these before when we were talking about the evolution of the eye. Flatworms have a little cup with these photosensitive cells at the base. They can't form an image, but the fact that it's a cup allows them at least to tell which direction the light is coming from. We have a gastrovascular cavity, just like we had in the cnidarians. Gastro meaning stomach, vascular referring to the vascular system, the cardiovascular system. There is no heart, but this cavity serves not just as a stomach, it also distributes nutrients throughout the organism. And you can see it's got all these fine branches that come off of it to do that. There's a pharynx, and it's not quite like the pharynx we're going to talk about in the next topic, but they actually have this really cool tube. It's almost like an elephant's trunk that attaches to the gastrovascular cavity. They can stick that out and they can grab onto things, usually little crustaceans and so on, bring them in and consume them. Okay, finally, on to the cestoidea. These are the tapeworms. The life cycle that you're seeing here is of a beef tapeworm. So we have a person that's eating some undercooked beef. I mean, if this is something you're really concerned about, all you have to do is get the meat up to 56 degrees for a couple minutes and that kills them. They're pretty easy to kill. But if you're having some really undercooked beef or maybe some steak tartare or something like that, maybe you will pick up a larval beef tapeworm and it will develop into the adult form in your intestine. We call the adult form, the mature form that can have sex, we call that the definitive form. It lives in the definitive host. Now, in a tapeworm, at the top end, we have hooks and we have suction cups, as you can see on the right there. That's a structure referred to as the scolex. There is no mouth, there are no eyes, there are no sense organs. None of that stuff is needed. All of that stuff has been lost because they live in the dark and they live in pre-digested food. At the other end of the animal, we have these segments called proglottids. Each proglottid contains an ovary and a testes. They quite often have sex with themselves. If there's another worm nearby in the intestine, they'll have sex with that, but they can have sex with themselves. These structures fill up with eggs. They're shed. They might end up on some vegetation. A cow will eat that. And then what happens is we have a larva developing from that fertilized egg. That gives rise to a larval form that will live in the meat. So this larva will burrow into the meat. It actually forms kind of a cyst, so it attacks muscle, and the muscle will kind of wall it off, and this little animal will form part of this wall as well. So we have this little walled off cyst, about the size of a grain of rice in the meat, and if you eat that, the larva will pass through your stomach. The cyst will protect it from your stomach acids, and when it gets to the intestine, the larva will burst out and give rise to the adult form. So there are two big groups of tapeworms that cause problems for humans. We have the beef tapeworm, that's the one we were just discussing, and we have the pork tapeworm. For tapeworms, there's always going to be multiple hosts. So we're going to have our definitive host, and that is where the tapeworm reaches sexual maturity, so where it reproduces sexually. And then we have an intermediate host. This will contain the larval form. So the sexually immature form is in the intermediate host. Now there's lots of other parasites out there. In many cases, they'll have several intermediate hosts. And in some cases, the larva can reproduce asexually. But the beef tapeworm and the pork tapeworm, their life cycles are pretty straightforward. They have two hosts, so they have an intermediate host, they have a definitive host, and there isn't any kind of reproduction that takes place in the intermediate host, and we have sexual reproduction in the definitive host. So for the beef tapeworm, the human is the definitive host. You cannot be an intermediate host. You cannot have the larval form if you're a human. The cow is the intermediate host. The cow will not be the definitive host. It will have the larva living in its meat. The larva is going to encyst in cow muscle. So it's going to form a little cyst around it that will protect it. And then it kind of just soaks up juices from the muscles that it's damaging. And that larval cyst needs to be ingested by a human from uncooked or undercooked beef, and then that larva can turn into an adult in the human. For the pork tapeworm, this is interesting, the human can be definitive or intermediate. So the human can have the adult tapeworm living in their intestine and shedding proglottids, or the human can have the larval form of the pork tapeworm living in muscle and tissue. And if you have a choice, it's better to be the definitive host. If you are the intermediate host for pork tapeworm, those larvae will be found not just in your muscle, but they can actually live in your brain, which causes a lot of damage. And they can even live in your eye. They can float around in the fluid of the eye, which is a disturbing idea. But if they're found in the brain, this is a pretty serious case. There's a number of people that are admitted to hospitals every year that are thought to have mental disorders of some sort, and they actually have these larvae living in the brain. If you have the larva living somewhere in your body, that's known as cysticercosis. So here again, a typical tapeworm. On the upper left, we've got the scolex of the tapeworm, so that's that bit at the top there. You can see them. The suction cups, those are not eyes. This is not actually a head per se, because there is no mouth and there are no sensory organs. And then you can see that the proglottids start forming right below the scolex, and they get larger and larger and larger as they move downwards. So on the bottom, you can see again a tapeworm, and the scolex, I'll just circle it, is this bit here. Each one of these big things that's being shed is a proglottid, and the proglottids develop in this direction. So they are initially made just below the scolex, and then they mature, and as they mature, they develop mature ovaries and testes, and have sex with themselves. So generally, one proglottid will send sperm to an adjacent proglottid. And then after that happens, the proglottids fill up with fertilized eggs. They're embryonated eggs, which means they start to develop slightly, and then those are shed. There's an interesting link there if you want to follow it. It's a page that discusses one man's tribulations, let's say, with a tapeworm. So it discusses a person that ate some steak tartare at a fancy French restaurant, got a tapeworm, and then he talks about how he got rid of it. So here's that beef tapeworm life cycle again, just very quickly. So we have this beef measle, which is an encysted larval tapeworm. And if you know what you're looking for, it is something that's visible. It's about the size of maybe a half grain of rice, something like that. And it's found within the muscle of the cow. If you eat that, you're taking in the larval form, and that will pass through the stomach. It's very resistant to stomach acid and will germinate, so to speak, within the intestine. Here's a close-up of the beef tapeworm scolex and also proglottid. Again, the scolex is not a head, even though it's at the anterior or front of the animal. There's no brain, no nervous system whatsoever. There's no mouth because there is no digestive system. There's no sensory organs. There's nothing that you would expect to find in the head of an animal. There's no respiratory system either. Behind the scolex, we have proglottids developing. They move downwards and mature, and they're shed from the end. And in the bottom right there, you can see a mature proglottid. It contains ovaries, it contains testes, and there's an opening. You can see it leading to the right side of the proglottid. That is a vagina. There's actually an opening for the sperm to swim through to get to the eggs, fertilize the eggs, and then each one of these segments will fill up with hundreds and thousands of fertilized eggs that start to develop into a very early embryo, and then the proglottids are shed in that state. So with the pork tapeworm, humans can also be an intermediate host. That's not the case with the beef tapeworm. Again, with the beef tapeworm, humans can only be the definitive host. They can only have the mature animal living inside them. Tapeworms, like all parasites, are very picky about the host that they infect and the stages that they can complete in that host. So with the pork tapeworm, let's say that you're a farmer, and let's say that you're breeding pigs, and let's say that those pigs have tapeworms in their intestine. So they have adult, sexually mature tapeworms living inside their intestines. That would make the pig the definitive host. It is going to be shedding proglottids that contain embryonated eggs, or partially developed embryos. Now imagine that you use that pig manure that they produce containing eggs to fertilize other crops. Maybe you use it to fertilize your carrots, and you don't wash off your carrots completely. You can ingest the egg, and then the egg will develop into the larva in you. You will become the intermediate host. That's a bad thing because, as discussed, you could have these larvae living in your muscle, but you could have them living in your eye or in your brain. Now, let's imagine another situation where the pig is the intermediate host. So maybe the pig ate the carrot that had the eggs on it. The pig, if it's the intermediate host, is going to have the larva encysted within its muscle and perhaps other tissues. Now, if a human eats that pork and doesn't cook it fully, they can get the larva, which will pass through the stomach and germinate from the cyst in the intestine and become the definitive form. So in that case, the human would be the definitive host. They would have an adult tapeworm living inside of them. They would be shedding feces that contain eggs. And this can be a problem in some parts of the world and on some farms where proper hygiene is not followed. You could have a situation where both the farmer and his family and the pigs are all infected. They're all definitive and intermediate hosts, and they're spreading it back and forth between each other. Because in many parts of the world, human manure is also used as a fertilizer. So here's some images of what it might look like to be an intermediate host if you're unfortunate enough to be so. We have neurocystocercosis, and this is where you have the little larva living within the nervous system. And it's more common than you might think. So about 50 million people worldwide per year have this issue. This is an image of the surface of the brain of a person who had a very bad infection. They had lots and lots of these larvae living in cysts on their brain, causing damage, extensive damage to their brain. And on the right, at the top there, we're looking at the forearm, and we can see these cysts on the x-ray. These contain larval pork tapeworms. And at the bottom there, you're seeing an x-ray of the knee. Again, those little things that look like large grains of rice. Those are cysts that contain larval tapeworms, and they're doing considerable damage. All right, so let's move past that nastiness and look at something, well, that can be kind of nasty too. Let's take a look at body cavities and how these body cavities develop. So I mentioned this before. This is one of the characteristics we can use to separate different groups of coelomates. Coelomates are animals that have a coelom, and a coelom, spelled C-O-E-L-O-M, is a cavity. It's a body cavity. Now there's two types of coeloms. At the bottom there we have a true coelom. Note that they've drawn a red line around the inside of the body cavity, and also it's continuous with the outside of the inner organs. This red line here represents tissue that's derived from mesoderm. So we have this cavity that's completely lined, the organs and the body wall, by material, by tissue that's derived from mesoderm. At the top there, we have a false coelom, or pseudo-coelom. And the example they're showing there is a nematode, a roundworm, an unsegmented roundworm. In this case, around the outside, we do have some stuff that's derived from mesoderm, and that would be the bundles of muscle that you can see. So there are some muscle fibers here that are derived from mesoderm, but the gut in the middle is not surrounded by anything derived from mesoderm. So we have mesoderm around the inside of the body cavity, but not around the outside of the organs. And we'll come back to that in just a moment and look at the nematodes, look at that pseudocoelom condition. But first, let's take a look at some true coelomates. So we have the mollusca. These are the mollusks. We have snails and slugs are basically just snails without the shell. We have clams and oysters. These are the bivalves. Bi meaning two. They have two valves, two shells. We have the octopuses and squids. And although these things don't seem like they're all that similar, they do share a common body plan. So yeah, there's quite a diversity. We have things like the snail down the bottom here. We have a scallop here. Those little dots are actually eyes, which is pretty cool. We have the nautilus, we have squids, we have cuttlefish, we have octopi. Lots and lots of species. Pretty much all of the big ones are marine, but there are, of course, some terrestrial ones like slugs, and we do have freshwater snails as well, and terrestrial snails. They're bilaterally symmetrical because they do not always have a well-defined head, but even when they don't, they do not have a radial symmetry to them. They're triploblastic. So we have our three layers, our three germ layers in the early embryo. They have true tissues, of course. They have well-developed muscles. They have a very limited circulatory system. It's an open system. I'll talk about what that means in just a moment. They have a one-way gut, so a flow-through gut. What I mean there is they have a mouth and an anus. Food goes in one direction. It goes in one place and out the other. And they do have a body cavity, although it's not quite as well developed as we see in other coelomates. This is the ancestral form of the mollusks. They all have a body form that's based on this in some way. They have a foot, so you can see that at the bottom, and most of them use that to slide along on. So something like a slug or a snail will have mucus on the bottom, and they can slide along that mucus. We have a visceral mass. So viscera means organs. So this is the big chunk in the middle there that contains all the internal organs, and they do have gills generally, and they have a fairly simple digestive system and a simple circulatory system. They have what's called a mantle, and that's the layer that's shown in purple here, and it secretes a shell. So even in things like octopus and squid, there is an internal remnant of that shell. They have a mantle cavity, and this is the cavity just beneath the mantle, and typically that contains the gills and also the anus. Now the other thing they may or may not have is something called a radula, and you can see a close-up of that there. This consists of little rasping teeth that are attached to a tongue that they use to scrape objects for food. So if you've ever watched a tropical snail, maybe in a tropical fish tank, feeding on algae on the glass, they have this tongue that they use to scrape the algae off and break it up into little pieces. Now I mentioned that they have a simple circulatory system. You can see we have a heart there, and it's attached to a tube, a short tube. The heart is really just a swelling, a muscular swelling of that tube that can contract and move fluid. But notice the tube doesn't go very far, and that's because blood, in this case, it's really just interstitial fluid. It's just fluid that can seep between cells. The quote-unquote blood is pushed from that heart down that tube, and then it just kind of seeps between the cells, and then it's recollected at the other end of the tube, and this just helps circulate stuff. It's kind of like a sump pump almost. So the groups that we'll look at We'll look at the gastropoda, which are the snails and slugs. We'll look at the bivalves, and we'll look at the cephalopods. The polyplacophora are quite fascinating. Those are known as chitons, and quite common in BC in tide pools. So they're quite often exposed at low tide. And they look a whole lot like the ancestral mollusk. So they have these plates on the back that overlap, and the actual animal looks a lot like a slug. So let's start with the gastropoda. Gastro means stomach, poda means foot, stomach foot. What it means is they crawl along on their stomach. So this would be things like snails and slugs. Again, slugs are just snails that have lost their shell. And we do have some organisms called nudibranchs, which are found in oceans that can be quite colorful and sometimes rather painfully toxic as well, hence the colorful nature. But these would also fit into this group. Next, we have the bivalvia or bivalves. Bi meaning two, they have two shells that clamp together. They're attached at a hinge. And we can see internally here they have a fairly simple structure, although more complex than anything we've talked about so far. We have a very small coelom, a very small body cavity, and that's why we're starting with these guys as the coelomates. We have a heart, which is really just a swelling on a short tube. We have the stomach, and we have a short esophagus, a short intestine. We have a foot, and the foot can be extruded between the two valves. So if you ever find a clam that's buried in the sand and you want to, abuse it slightly, you can pull it out of the sand, and then it's really fun to watch it bury itself back in. It'll stick that foot out, it sticks the foot into the sand, it expands the foot, it pulls on that, and it pulls itself back into the sand. These are filter feeders. So what they do is they will take in water, and they take that through this in-current siphon here, and the water will go over the gills. So they're extracting oxygen and they're getting rid of waste carbon dioxide. But at the same time, the gills are very, very sticky. They're covered in mucus. They've got cilia as well. And what the cilia will do is they'll move that mucus back towards the mouth of the animal, which is over here. Now, once the animal has digested that food, then what's going to happen is the food, of course, will go through the digestive tract and come out the anus here, and then it's expelled with the outgoing water. So waste carbon dioxide, waste food is going to leave through the anus. And what you're seeing on the right there is a very young little clam. Another thing that's really quite fascinating about freshwater clams is that most of them have a parasitic form. So the very, very early clam that's shed out into the water from an adult female clam will grab onto the fins of a fish and actually suck blood from it for a while. These are really tiny, they're just barely visible, and then eventually they fall off. So really interesting system. That's how the early clam gets its first food. It's also how the clams are dispersed throughout the lake. In the middle here, we're seeing a scallop, and down the bottom there, a scallop again. And some scallops do have little eyes that are light sensitive. They're not really forming much of an image, I don't believe, but they are light sensitive. And of course, in oysters and clams, we can have the production of pearls. And what's happening here is we have a bit of irritant, like a grain of sand, that's trapped under the mantle. The mantle produces shell, and it also produces the mother of pearl, that very, very smooth substance that lines the shell so that the internal organs won't stick to it. Well, it covers this irritant in layers and layers and layers of mother of pearl. And that gives us pearl. And on to the cephalopoda, the most advanced of the mollusca. So cephalo means head, poda means foot, so these are head-footed mollusks, I guess you could say. So what that means is that the foot, that muscular, fleshy foot, has become the tentacles and the head, the portion of the head that supports the eyes. And the eyes are very well developed. So for an octopus, their vision is probably pretty much on par with our own. They're also the most intelligent of the invertebrates. So the cephalopods can be highly intelligent. You might have seen videos of octopuses that can open up a jar, for instance, to get to a crab that's inside. So very, very highly evolved mollusks. The foot, once again, has become the tentacles and part of the head. In the cuttlefish and in the squid, the torpedo-shaped part is derived from the mantle. Okay, we'll leave the mollusks now and take a look at worms, specifically annelids. So I mentioned before that the term worm is not a great term because a worm is simply something that's much longer than it is wide. And there's many things that would constitute a worm. So we have the annelids that we're about to talk about. We have the nematoda, we have the platyhelminthes, and some other oddballs. But the annelids are round in cross-section, and they're also segmented. So these are the segmented roundworms. Now, many of them have little appendages, and even an earthworm would be an annelid. An earthworm has tiny little bristles. You might not realize that. You have to look at them very close. But if you take an earthworm and clean off the dirt and take a look at it with a compound microscope or a magnifying glass, you can see these little bristles that help it stick to the soil. So they help it grab onto the soil. There are other worms that have much longer bristles. They have these little appendages, almost look like legs. They're not jointed, but these large bristles that do function somewhat as legs. And finally, there's a third group that doesn't have any of these bristles or quote-unquote legs, and instead has suction cups, and those are the leeches. And we'll look at each of these. So this here is an example of an earthworm, and here we have an example of something called a polychaete. So earthworms belong to a group called the Oligochaeta, which have little bristles. We have the polychaetes, which have big bristles that look like little legs, and then we have the leeches that don't have any bristles or appendages whatsoever. There's at least 15,000 species. Lots of them are marine, but of course there's some that live in the soil and there's some that live in fresh water as well. They're bilaterally symmetrical. They have 3 tissue layers. They have true tissues, of course. They have a closed and complete circulatory system. Now that's different from the mollusca. So compared to the mollusks, they have a much better developed coelom. It's much larger, so the body cavity is much bigger. And also their circulatory system is more developed. They still don't have a heart with several chambers to it like we have, but the blood is always traveling through little tubes. That's what the closed part means. In the mollusca, the blood flows along a tube for a little while and then it just seeps between the tissues. That's an open system. They have a one-way gut, so they have a mouth and an anus. And like I said, they have a well-developed body cavity. So our 3 classes in this phylum. We have the Oligochaeta, and this would be the earthworms and their relatives. There's some freshwater forms as well. They have single setae. Don't worry about that term, but that's the term for the bristles. So they have these bristles on each segment. There are aquatic species, as mentioned, but there are, of course, a lot of terrestrial species. And here's a typical one here. That weird structure in the center here towards the front end, that's something known as the clitellum, and that's used in the manufacture of cocoons. We have the polychaeta. So the name literally means many setae, many bristles, but the bristles are much larger. And they're referred to as parapodia. So they're kind of like little legs. And they can act as feet, and they also act as gills. So they can be used to extract oxygen from the water. They're marine, but there are some freshwater ones as well. They're free living, so they're not parasitic, but they may build a tube that they live in. And this is what they look like here. This is one that has these well-developed gill-like structures that it sticks out from the sand, and it uses that to extract oxygen. Some of these guys have quite a nasty bite, actually. Unlike the earthworms, they tend to have a fairly well-developed head with a well-developed mouth and eyes. There are many of them that will burrow into the mud, and you can find them at low tide, but I wouldn't recommend you grab them because, like I said, some of them bite pretty hard. And then we have the leeches, and just know them as leeches, that's fine. We don't have any setae, we don't have any bristles whatsoever. We have a reduced coelom compared to the others. They're freshwater, and they can be either predatory or parasitic. If you go up to 10 Mile Lake, there's a lot of really pretty leeches there. I find leeches quite interesting, actually. But most of the ones you'll find are actually predatory, and they're hunting little crustaceans and so on. But of course, some of them are bloodsuckers as well. They tend to take really good care of their young too. That's one thing that's kind of interesting. So if you find a leech in the spring or early summer, have a look at the underside of the leech. There's a good chance there will be a bunch of little babies attached to her. They carry their babies around for quite a while. They produce a substance that is an anticoagulant, which has proven to be rather useful in medicine. They also produce a local anesthetic. So when they bite you, they temporarily will take away feeling in that space so that you don't know you're being bitten, and they release this anticoagulant that stops the blood from coagulating. So of course they used to be used in medieval times for bloodletting, for getting rid of bad blood. We don't do that anymore, but we do. We have taken some rather important compounds from them, and also they are used to promote blood flow to limbs that have been reattached. Let's say you lose your finger, they have to stitch it back on. They might actually put leeches on it to create more blood flow to that limb, which will help with the repair of blood vessels. All right, so again, we have the Eligakida, reduced head. So again, if you look at a worm, it's kind of hard to tell which end is which. You've got that clitellum, that's going to be closer to the front. With the polychaetes, a very well-developed head with little eyes, and like I said, sometimes a very nasty bite. And then we have the leeches, which have a flattened body, reduced coelom. The segmentation is clear, but there's no bristles. There's no bristles of any kind. So looking at those one more time, the Eligakida, the most common example would be the earthworm. You can see at the front of the earthworm, there are structures there labeled pumping vessels, so heart, basically. Those are really, really simple hearts. There's several of them, but it's just a swelling, a muscular swelling of a tube, but they do have multiple hearts. Unfortunately, you cannot cut an earthworm. You can't cut its head off and have it regenerate a head. That's a popular myth, but it doesn't work with earthworms. It does work with flatworms. We have the polychaetes here. Again, you might see these at low tide. The one that's in the upper left there is something you might find in the mud at low tide. There's a number of very colorful ones that you might find at the bottom of the ocean as well. And some of them do have these feathery appendages that they stick out. Usually these ones live in a tube, so they will produce this tube and then stick out these feathery appendages that they use to collect oxygen. And that's the head of one down the bottom, so you can see the well-developed eyes. And then we have the leeches. Leeches tend to have two suction cups. They have a big one at the back end, and they'll use that to stick themselves to a rock. And then the front end will kind of wave around looking for a host that they can latch onto. And there's a small suction cup there next to the mouth. that they'll use to grab onto your leg, for instance. Now, they're ectoparasites. That means they're external. They don't have any internal larval stages or anything like that. But yeah, many of them are bloodsuckers, but many of them are also just hunters. They're predatory. And here you can see a mother leech with her little babies. So some of them will produce a cocoon-like structure and just leave that stuck to a rock or stuck to the underside of a water lily or something like that. But some of them are really good mothers, and they'll carry the babies around for quite a long time. It's actually kind of cute. Apparently, acupuncture with leeches is a thing. I don't know about the efficacy of that, but as mentioned, we do have anticoagulants and also local anesthetics that come from leeches. And now on to the nematodes. These are non-segmented roundworms. So again, if you look at them in cross-section, they are round in cross-section, but they do not have segmentation. They also present a bit of a problem for taxonomists. They are pseudo-coelomates. So that means they don't have a true coelom. The outside of the coelom, the body wall, is lined by muscles that are derived from mesoderm, but the outer covering of the internal organs is not. So the body cavity is not completely lined by derivatives from mesoderm. Now, traditionally, they were placed with the rotifers, which is not a group we're going to talk about, but these are kind of interesting little microscopic critters. They're freshwater. You find them in ponds and things around here, and they also have a pseudocoelom. So that's on the right. You can see a tree that's based on just the body plans. However, using molecular data, the nematodes appear to be closely related to the arthropoda. The nematodes do share one interesting behavior and physiological character with the arthropoda, and that is they shed. So they have a cuticle, they have this outer tough layer that they molt. And the technical name for molting is ecdysis. So they're referred to as the ecdysozoans, along with the arthropoda. So odd that these little worms would be closely related to insects, but based on mostly molecular evidence and also the fact that they molt, this would appear to be the case. So it's the tree on the left that we're going to be using. So once again, for a true coelomate, we need to have derivatives of mesoderm lining the entire coelom, the entire body cavity. So that includes the inside of the body wall and also the outside of the organs that are in that cavity. And that's not something we have in nematodes, although it's thought that maybe nematodes had this and then lost it. So it's a secondary characteristic that appeared later. Nematodes are very common. They're found in marine environments, they're found in freshwater environments, they're found in the soil. Most of them are microscopic, but there are some parasitic forms. As mentioned, they are ecdysozoans. Ecdysis means to molt or shed, so they do have an exoskeleton that they shed. They're roundworms because they're round in cross-section, and there's lots of species, at least 90,000. These are species that have been identified, probably a lot more than that. As mentioned, most of them are microscopic, and they do look pretty similar. They vary quite a lot in size, so they can be 1 millimeter or even less. The big ones, which are always parasitic, can be up to 1 meter in length. So there are some that are parasitic in whales, for instance, that live in whale intestines that are quite long. They're bilaterally symmetrical, they're triploblastic, of course, they have true tissues, they have well-developed muscles, they're arranged longitudinally, so they run the length of the animal so that its body can kind of thrash side to side, kind of like an eel, so that's how they move. They have one-way gut, so they have a separate mouth and anus. They have a reduced coelom, and it's also a pseudocoelom. It's not a true coelom, and in some species it appears to be lost. So this pseudocoelom is probably a secondary characteristic. They don't have a circulatory system or excretory system. As mentioned, most of them are quite small, and the small ones really don't need a system to get stuff in and out. They can just work by osmosis. And the parasitic ones are generally living in tissue. They're feeding off of fluids within the tissue, or they're living in the digestive system and the food has been digested for them. And it's pretty easy to get materials in and out. Now, having said that, they do have a digestive system. They do have they do have reproductive organs as well. So this is a microscopic one. On the outside, we can see the cuticle, and the cuticle is very tough. This is the exoskeleton. It's got ridges on it that will help it grip material and move around. This is a species that would normally be found in fresh water or in the soil, but it can also be grown in the lab really easily. It consists of a little over 1,000 cells, and a lot of what we know about development comes from studying these tiny little worms that are about 1 millimeter in length. You can actually grow them on Petri plates. It's quite fascinating because every single cell has been mapped in this particular species, and we know where every single cell comes from. So they've actually studied right from the zygote to the fully formed worm and traced the fates of every single cell. And that's what they look like under a light microscope. The top image there is under a scanning electron microscope. All right, moving on to the largest group, the Arthropoda. And again, these are members of the Ecdysozoa because they undergo ecdysis, which means they molt. So they have an exoskeleton, an external skeleton that they would shed. Lots of groups here. We'll focus on the ones that are highlighted in yellow, but I'll just mention some of the others as well. We have the trilobites, which are entirely extinct, but they were a very, very common group. So they went extinct with the Permian extinction, which was the largest of the extinctions. And yeah, they're totally gone, unfortunately. We have the chelicerates, which include horseshoe crabs and scorpions and ticks and spiders. We're going to just focus on the arachnids, the spiders from that group. We have the Uniramians, which is the insects, millipedes, and centipedes. And we will look at each of those classes, but I don't expect you to know the term Uniramian. And then we have the crustaceans, which are crabs and lobsters and shrimps and even barnacles. Barnacles are really unusual. So we have this shell. It almost looks like a bivalve, but inside that you've got an upside down crustacean with its feet sticking out, and it's using its feet to grab onto food particles to filter food out of the water. So most of the arachnids are terrestrial, although the larger group does include horseshoe crabs as well. We have a cephalothorax, so cephalo meaning head, thorax referring to the chest. So we have these two parts that are fused together, and there are 6 pairs of appendages, so 12 limbs. The most anterior appendages are called chelicerae. So these are modified from legs. We've talked about that in class a bit, how all of the arachnids, all of the arthropoda, they start out as embryos, as something that looks kind of like a centipede. But then the frontmost limbs become things like chelicerae. They become part of the face. And if you take a close look at an arthropod and look at its face, there's a lot going on there. There's lots of little modified legs on their face. The chelicerae are tiny little pincers, and you'll see those on scorpions, for instance, or fangs in quite a few of these groups. We have pedipalps. You see those on spiders. They're quite pronounced. They do look like little legs on the face, and they're used to as kind of sensory structures to kind of sense a food item and also to manipulate it as well. Then we have 4 pairs of actual walking legs. So again, we have our chelicerae, little pincers or fangs that make up part of the mouth. We have pedipalps, which are almost like tiny little antennae, but they're on the face. And then we have 4 pairs of walking legs. We have an abdomen. So we have 2 body regions. We have the cephalothorax, and then we have the abdomen. They have what are called book lungs, which are interesting structures. They do look kind of like a book. I'll talk about that in just a second. So here's some examples. Here we have a scorpion, we have a spider, and what you're seeing in the other image there is spider silk. Spider silk is incredibly strong. For its width, it's stronger than steel. So there's lots of applications for this, for making sutures, for instance, and perhaps at some point even making clothing, making like Kevlar type clothing. So here we've got a section through a spider, and we can see some of the bits and pieces. Down the bottom there you can see the book lung, and it's basically like pages of a book. So you've got this structure where there are these thin structures that look like paper from a book, like leaves of a book, and that increases surface area. Air is taken in through little holes on the surface. It's passed over these pages of the book. These pages are moist, and then that's going to extract oxygen and get rid of carbon dioxide. So some other examples here. We have at the bottom there kind of a typical scorpion. At the top there we have what's called a pseudo scorpion, and these are very tiny. They're not quite microscopic. You can see them with your eye, but you'll find these quite often in compost heaps, so something to look out for. You can find them in soil. They are found in Canada. We've got a tick here, which would also belong to this group. We've got a black widow spider, of course. This is a crab spider. Kind of cool. They kind of look like crabs, but also they move sideways. They tend to hang out in flowers like this one is doing here. And if a bee comes to the flower, they'll jump on it. So instead of spinning a web, they actually will run around and hunt and tackle their prey. And then down the bottom here, we have a wolf spider with lots of little tiny babies on its back. I remember once I was driving in Oklahoma. It was in the summer, and I was going down the road with high beams on, and these spiders can be very big, and I saw one run across the highway, and I could see all the eyes of the little tiny babies on its back. It was kind of creepy. Next, we've got the Diplopoda. Di meaning 2, poda meaning foot. These are the millipedes. The di refers to the fact that for every segment they have 2 pairs of legs, so they have 4 legs. They're terrestrial. They feed on decaying plant matter. So you don't have to worry about these guys. They don't bite. Some of them can be poisonous. Some of them are brightly colored because they're poisonous, but only if you eat them. So just don't eat them and you'll be fine. The name millipede means 1,000 legs. They don't have that many, but they do have lots. The other group that's closely related is known as the Chilopoda. Incidentally, if you see CH together, especially at the beginning of a word, probably means it comes from Greek, and the CH is a hard K in Greek. They have antennae on their head. They have 3 pairs of appendages that make up the face and mouth parts. So they have mandibles, they have poison claws, so they have these modified short little legs that are poison tipped. And the Chilopoda, the centipedes, are poisonous. They do have a poisonous bite. Now, centipedes won't bite you unless you grab them, typically, but they can bite, and it can be quite painful. They have 1 pair of legs per segment. They're terrestrial. They're carnivores. They hunt. And yeah, they're commonly known, as the centipedes. Centipede means 100 legs. They typically don't have that many. They typically average around 40 or so. But here's a look at a tropical centipede, one of the few creatures that really kind of frightens me, I would say. That and sharks. And you've got a drawing there that shows the face from the side, and you can see at the front there these little limbs that are actually the poison claws that they use to poison their prey. They work like fangs. So here's a nice image of the face of a millipede, and you can see the compound eyes. Looking at the underside here, you can very clearly see that there's 2 pairs of legs, so 4 legs for every segment, and that's the defining feature of the millipede. And like I mentioned, some of them are brightly colored. It's to warn something that might eat them, like a bird, that they don't taste very good. They have mild toxins in them. But they do not have poison claws, and they're not carnivorous. And the centipedes, the Chilopoda. So here's a nice image, a creepy image, of a tropical centipede. They can get quite large, and you can see that they have 1 pair of legs, so 2 legs per segment. They have 2 kind of backward facing legs as well, the 2 that are coming out the posterior there. And these guys do have quite a nasty bite, but they are typically quite skittish. Centipedes tend not to bite unless they feel threatened. If you, I don't know, roll over on one or grab one. Some of them are quite good mothers. You can see an adorable picture here of a mother centipede and her babies. And this picture here is kind of creepy. It's an interesting story too. This guy was living in London, England, and he had ordered some furniture that came from a tropical country. He set up the furniture and he could hear something scurrying around the room, and much to his dismay, he pulled this out of his sofa. Moving on, let's look at the crustaceans. So they are freshwater and marine, mostly marine, and there are a few terrestrial species as well. There's at least 40,000 different species of these. Some of them are really small. So we have some freshwater crustaceans known as Daphnia, or water fleas, that are quite small, and they're found in the plankton in lakes and ponds. They have 2 pairs of antennae, and they have appendages attached to the thorax and also the abdomen. They have gills for respiration, the big ones. Some of the little ones don't actually need them. And they have a rather tough exoskeleton or carapace that's made of calcium carbonate. And here's some examples. So we have some ghost shrimp here that are actually on some coral, and we have a lobster. These are the terrestrial ones. So we were talking about these. They have so many different names. I know them, coming from Ontario, I know them mainly as woodlice or pillbugs, but some people call them roly-polies. Some people call them penny pigs. And if you go further east, you go to the east coast, they call them carpenters because they tend to live in rotting wood. And let's now take a look at the insecta, or hexapoda. Hexa means 6, poda, legs. This is by far the largest group of animals in general. So about 75% of the species of animals on the planet are insects. There's at least 2 million species of insect. So they're found in every terrestrial environment. There's 32 separate orders of them. We have really good fossils going back more than 400 million years. They can move around just about any way you can think of. So a lot of them fly, a lot of them swim, they crawl, they hop, etc. And they have typically this 3-part structure to them. So they have 3 divisions of the body. They have the abdomen, they have the thorax, and they have the head. Of course, if we're talking about larval stages, that might not quite be the case, but in the adult, they have, again, the abdomen, thorax, and head, and the appendages are attached to the thorax. So they're going to have 3 pairs of legs, 6 legs, attached to the thorax, and then also maybe a pair of wings, maybe 2 pairs of wings. They have a tracheal system for respiration. So they don't have lungs like we do, and they don't have book lungs like we see in the arachnids. Instead, they have these little openings known as spiracles, and they open into tubes known as trachea. And the tubes will conduct air directly to the tissues. So they have a ductwork They have quite literally like an HVAC system in their bodies that carries air to all the tissues. They have a ventral nerve cord. Now, what do I mean by that? Ventral means towards the stomach. Nerve cord is just referring to their simple nervous system. So you can see on the bottom figure there, the yellow cord down the bottom, that's the nerve cord, and it's ventral. Now, why is this important? Well, note that they also have a dorsal artery. So at the top there, we have that red line, which is the main artery in their body, and it runs along the back. Dorsal means back. If you take a look at vertebrates like ourselves, this is flipped around. We have a nerve cord that runs along the back, that's our spinal cord, and then our major artery runs towards the belly, towards the ventral surface. Some people have hypothesized that the first vertebrates were essentially upside down invertebrates, but we'll talk about that later. This is, like I said, the largest single class of animals, at least 1 million species. There's 1 million species that are documented, but there's a lot of people that think there might be 2 million or more. So the Insecta, like I said, really good fossil record. Back in the day, Mississippian times is the time period, there were dragonflies that had 4-foot wingspans, which is pretty amazing to think of. However, insects kind of learned that being small and numerous was a better strategy than being big and all the problems that come with being big. Here we've got a stick insect. So it looks like a stick, and it would live up in the trees. In areas of the southern US where there's lots of these stick insects, you can walk into the forest and it sounds like it's raining, and it's actually eggs just being shed by these guys up in the trees. This is a really interesting one. So at the bottom of this pit is something called an antlion, and this is the larval form of a flying insect, but the larva lives at the bottom of this pit. And ants will fall into the pit. This is what it looks like. It's pretty scary looking. So the jaws that you see at the front there, they would stick out at the bottom of this pit. Ants will fall into the pit. If they don't, the antlion will actually throw sand at them to knock them down, and then, of course, it sucks all their juices out. That's what it looks like there, and there's the adult. There's lots of cool insects that are active in the winter. What you're seeing here in the bottom right is something called a springtail. These are really tiny, and you'll see them on the snow, especially if you're cross-country skiing. They tend to like cross-country ski trails. I don't know why, but you'll see little black dots on the snow, and if you look really carefully, you might see that they're moving. They're these little tiny insects. They have this little spring mechanism, so that thing that's sticking out the back looks like a tail. It's actually kind of a prong that can be ejected very quickly, and they can hop very, very far. Now, this is one of my favorite quotes, maybe. There was a very famous entomologist, that's someone who studies insects, and he was asked at one point, I'm not sure by who, but he was asked by an interviewer, what has the study of biology taught you about the creator, Dr. Haldane? And his answer was, I don't know anything about God, but he's obviously inordinately fond of beetles. Yeah, beetles are the largest group of insects. So insects are the largest animal group, and of that, beetles are the largest group. We have at least 250,000 species of beetles. I quite like that little drawing in the middle. These are dung beetles that would normally collect dung into little balls on the African savanna and then bury that and lay their eggs in it, and their larvae would eat the dung. But here they are moving the world because, well, they kind of do. Okay, so here's the groups that we're looking at for the arthropoda. Arthropoda, I don't know if I mentioned this, but it means jointed leg. Think of arthritis, that's an inflammation of a joint, but they have these legs that are jointed, that are made up of an external exoskeleton. Anyway, we have the arachnids, we have the diplopoda, we have the chilopoda, we have the insecta, and we have the crustacea. Okay, so we'll wrap up here by taking a quick look at the cladogram for these invertebrates we've just talked about. At the bottom here, we have the porifera, the sponges. This is the parazoa. They're not quite true eumetazoans. They're not quite true animals, and I put them as being multicellular here. They're on the cusp of being multicellular. They don't really have much in the way of tissues, but we do have a close relationship between the cells. Next, we have the first group of true animals, the Cnidaria. These are definitely multicellular. They do have tissues. They're pretty simple, but they do have tissues. They are diploblastic. That means they only have 2 tissue layers. And going back to the embryo, they only have 2 germ layers. They never develop that mesoderm, and they don't have any derivatives of mesoderm. They have a gastrovascular cavity. And remember that they do not have a one-way or flow-through gut. They have one opening which serves as the mouth and also the anus. Next, we've got the platyhelminthes. Those are the flatworms. Most of them are free-living, but some of them are parasitic. Some of them do have a simple nervous system, especially the free-living ones, because they need to coordinate movements for hunting and so on. But the parasitic ones, remember, have lost all of that stuff. The cephalization, we see that in the free-living ones. In the tapeworms, we don't have a head, and cephalization refers to the evolution of a head, but almost certainly the parasitic forms did evolve from something that was cephalized. They evolved from flatworms that were free-living and more complicated, and then they lost all the complexity because they didn't need it. Next, we have the phylum Nematoda, the nematodes. Where we put them depends on whether we follow just simple body plans or whether we follow molecular studies. So I'll come back to them after we look at a couple more groups. We've got the mollusks, which have a true coelom, but it's quite reduced. They have a circulatory system, but it's very simple, and it's open, which means the blood spends a lot of time just seeping between tissues instead of traveling in tubes. We have the annelids, which are the segmented roundworms. They have a true coelom. The coelom is filled with fluid. So for an earthworm to move, what they do is they contract muscles, and that squeezes one of those compartments, one of those segments, and the fluid that's inside the segment pushes back. So they have what's called a hydrostatic skeleton. They have a totally closed circulatory system, so that's quite a bit more advanced than what we see in the mollusks. We have the arthropoda, which are segmented, and these segments are repeated over and over again. So we have, for instance, within the insects, we have 3 body regions. We have the head, thorax, and abdomen, but within each of them we have several separate segments. They have well-developed appendages, most of them. They might not have that in their larval form, but most of them have well-developed appendages, and they have an exoskeleton. They have an open circulatory system, which is interesting though. So the heart is a simple structure. It's basically just a swelling on a tube that's muscular, and the blood will seep out and seep between tissues. Interestingly, in the arthropoda, the blood comes in many different colors. It's not always red, although it is sometimes, but in a lot of them it's green or even purple. Now, going back to the nematoda, we can place them between the platyhelminthes and the mollusca if we go by the old system, which is just looking at body plans, looking at body structure. So they are pseudocoelomates, and based on the fact that they have a false coelom, we might put them between the platyhelminthes, which are acoelomates. They don't have a body cavity at all, and the mollusca that have a simple body cavity. So that's one way to go. But based on more modern molecular analysis, they do seem to be closely related to the arthropods. They do undergo ecdysis, molting. They do have an exoskeleton, unlike the other worms we've talked about. And the pseudocoelom might be a secondary characteristic. It might be something that popped up later. And then finally, at the end here, a group we haven't talked about yet, we're going to talk about next, is the phylum Echinodermata. These are deuterostomes, and that's why we haven't talked about them yet. So they are closely allied with chordates like ourselves. And here's our terminology. And please do take a close look at the guide that I put for the non-deuterostomes and deuterostomes. That'll give you a sense of how much detail I'm expecting you to know.